Hi, my name is Nihar Patel, and today I'm going to talk about my paper, which is entitled Identification of Suitable Reference Genes for Real-Time Quantitative PCR Analysis of Hydrogen-Treated Human Umbilical Vein Endothelial Cells. So, a brief background: certain conditions the body generate reactive oxygen species, which in turn leads to tissue damage. These oxidative species, which causes oxidative stress, also affect gene expressions, and gene expressions can be quantified. So, quantitative real-time PCR application. To quantify overexpression, we need to compare it to a reference gene. Since no single gene is expressed constantly across all cell types and pathological conditions, poor reference gene selections will yield inaccurate gene expression results. A solution is to use a candidate gene that is stably expressed under varying conditions. Hence, the goal of this paper is to find suitable candidate reference genes for quantitative, real-time PCR analysis of hydrogen peroxide treated endothelial cells. So the methods. They took human umbilical vein endothelial cells, or HUVECs, and they cultured it with varying conditions of hydrogen peroxide for 24 hours. They then extracted the RNA from these cells, which is then reverse transcribed, and the quantitative real-time PCR technique is used on it with 15 unique test primers. The 15 reference test prime genes or test primers used, they were used because they're most commonly used in the field. So a brief background in quantitative real-time PCR. It works by using reverse transcriptase to add a complementary strand onto mRNA. Reverse transcriptase then digests the mRNA and adds a second complementary strand onto the first. This forms a double-stranded DNA molecule, which then goes through PCR as usual with a fluorescent marker that releases light depending on the amplification rate. The amount of fluorescence directly relates to the amount of amplification occurring, and this is plotted over time. The benefits of this technique is that it requires a low input template and is highly sensitive and specific. So they also use statistical analysis. They use two statistical analysis packages to evaluate the stability of these genes. In order for them to work, CT values were converted to relative quantity using the following equation, which is shown on the slide. The first package was called GeNorm. This analyzes the stability on average pairwise variation of one gene against all control genes, obtaining an M value. The lower the M value, the higher the stability, and vice versa. The second package was the Norm Finder. This estimates inter and intra-group expressions variations and calculates a stability value. The lower the stability value, the more stable this reference gene, and vice versa. So expression profiles of reference genes. The image on the left represents the CT values of all 15 candidate reference genes, while the image on the right shows all the statistical descriptions of these CT values. CT values range from 9.225 to 36.19, representing a wide variation. 18S was the highest expressed and had the widest range, while PUM1 was the least expressed, and ALAS1 had the least range. So genome analysis. Running the genome analysis and sorting the results yields this graph. As you can see, they made it somewhat linear. Remember that a lower M value means most stably expressed. Based on this, the most stably expressed genes are the U6 and TFRC. The least stably expressed is the 18S. They then use Norm Finder analysis. Running the Norm Finder analysis and sorting the results yields this graph. A lower stability value indicates a more stably expressed gene. Based on this, ALAS1 is the most stable, while 18S is the least stable. Comparison of analysis ranking. This table shows a ranking of each statistical analysis from the most stably expressed gene to the least. Although it is not a perfect match, 4 out of 5 of the top 5 ranks are the same, and 7 out of 7 of the lowest ranks are the same too. So we can say this analysis is somewhat accurate. So in discussions, this technique allows for the normalization of genes of interest against the control. Because of the way quantitative real-time PCR works, CT values which are high or low are undesirable, as they would provide an inaccurate result, making 9 out of our 15 reference genes suitable for this type of testing. Stability changes under different conditions, thence commonly used reference genes such as 18S and GAPDH aren't ranked very well in this study. However, only RP, LPO, and TFRC are correctly represented as being suitable by both this study and by common use. So in conclusion, rankings of stably expressed genes may differ based on the statistical program used, though both NormFinder and GNorm found mostly the same 5 stably expressed genes. Therefore, the best reference genes for the use of hydrogen peroxide treated UVEC cells are U6, TFRC, RPLPO, GAPDH, and ACTB. Thank you.